Okay, so continuing on with enzymes. So they work best in a particular temperature, a particular environment, pH, so on. All right, so then some enzymes, getting a little bit more technical here, some enzymes require something else in order to work appropriately. And that something else are often called cofactors. So co in front of something means like together, with, and so it's something that works together with an enzyme and helps the enzyme function properly. So cofactors are non-protein helpers. So they help enzymes to work, but they're not proteins themselves. So examples of those are minerals. Sometimes they can be metals. Zinc, iron, copper are examples of those. So therefore, enzymes, in order to work properly, be in its right shape, to be able to catalyze the reaction, needs to have these things. And so therefore, if you don't have those things, then the enzyme's not going to work properly. So they're cofactors. And then there are some substances also called coenzymes. These are like, they behave just like cofactors, except they're organic, meaning that they contain carbon. So a lot of our vitamins are organic molecules that help our enzymes to work. So therefore, like B12 will help an enzyme to work. And so therefore, if you're deficient in a particular vitamin, you have side effects because certain enzymes that those vitamins help to work properly do not work properly. All right, so those are some other environmental factors, or not environmental, but things in the cell or whatever that is going to affect the functioning of an enzyme. Another thing that can affect the functioning of an enzyme, besides the environment and cofactors and coenzymes, are what are called inhibitors. And just like the name implies, they're going to inhibit or stop an enzyme from working. So there's two different kinds. There's a competitive inhibitor, binds to the active site of an enzyme and therefore competes with the substrate. So in this, if we look at the next page here, whoops, I have another sheet here, turn this over. So this is the normal binding of the substrate with the active site. So remember the active site, there's a special spot in the shape of the enzyme itself that only the substrate can fit into, so it's the enzyme is specific to the substrate and they fit into that. Sometimes though you can have an inhibitor, so this right here in orange is the substrate, and this fits into the active site. And so sometimes an inhibitor can actually bind to part of the active site. So it might not have the exact same shape, but it might have the shape that matches part of the active site. And so therefore you have this competitive inhibitor. And now this little guy, this is your substrate, right? This is your substrate. If the competitive inhibitor gets there first, then the enzyme cannot bind, and therefore, even though you have the the substrate can't bind to the enzyme, even though you have the substrate technically present, it's rendered useless because the competitive inhibitor got there first. So they're competing for the same active site. So can you see that if the substrate gets there first, then the reaction will happen, but if the competitive inhibitor gets there first, then it won't happen. And so that's competitive inhibition. The second kind of inhibition or inhibitor is called a non-competitive inhibitor. And in this case here, here's your substrate again. All right, couple things happen. I'm going to get a different color here. Let's get a light purple here. All right, so here's your non-competitive inhibitor. All right, so the first thing that happens, all right, here is the non-competitive inhibitor. I'm just going to abbreviate here, inhibitor. Binds to a site, different than the active site. All right, so it binds to a site different than the active site. This is called, so it's, in this case here, it's on the opposite side of that. And so then the second thing that happens, so that's binding to that. So look at what happens to the active site. So this changes the shape of the active site. And the substrate no longer can bind. So it's the same, so it's inhibiting this enzyme from working in a different way by changing the shape of the active site. So you have the substrate here, but it just is not going to fit into the active site any longer. And so if we go back to this here, the non-competitive inhibitor binds to, and it's another part of the enzyme, that's what we just saw. And then the enzyme, which causes, so I have an arrow here, so that causes what to happen? Enzyme changes shape, and the active site is non-functional anymore. So therefore, the substrate can no longer bind. Okay, so so those are two types of inhibitors. So we have environment. Is important for enzymes and can regulate whether or not the enzyme is going to work properly. Cofactors and coenzymes, and whether or not there's the presence of an inhibitor. And so one other thing, and this is the last part of the chapter that we need to talk about, is how enzymes are regulated. So therefore, certain reactions you can be turned on and turned off, because not always do we need a chemical reaction occurring. So we have to have a way to be able to say, okay, we need this reaction to occur right now, but now we need to stop it. And so how does that happen? And so we can regulate that by regulating enzyme activity. So to regulate metabolic pathways, the cell switches on or off the genes that encode specific enzymes. So we can actually switch on and off genes. So we're not always making enzymes to catalyze every single reaction. So that's the first thing. The second thing is this thing called allosteric regulation. It says proteins' function at one site is affected by the binding of a regulatory molecule to a separate site, and the separate site is called an allosteric site. So we have several types of molecules that are regulatory molecules. One that stabilizes the active site or makes the active site stay in its functional shape. And so we call that molecule an activator. And then we have another molecule that stabilizes it in the inactive form, and that's called an inhibitor. So it makes the enzyme not work. So when the stabilizes the active site, so this is the enzyme is functional. If it's stabilizing in the inactive form, the enzyme is not functional. Okay, so let me illustrate with this picture below. I'll come back to this bullet here in a second, but I want to illustrate with this picture below this idea of activators and inhibitors. All right, so we're going to do some color coding. All right, so first let's just look at the enzyme itself. All right, so this is the enzyme in the active form. So we have active sites. So notice here it has 1 of 4, it says. So this is one. This is a protein. This is an enzyme that is in the quaternary structure. So this is one polypeptide chain subunit. This is another. This is another. This is another. So therefore there are 4 active sites. So let's color code that so we see what the active sites are. And then I'm going to do orange here. There's a regulatory site right here, or let's put here allosteric site, because we just wrote there, allosteric site. So here we have these guys here. All right, so those are your allosteric sites. So that's what we said here. It's a site separate than the active site. So that's what we're looking at. And so, so look at what happens when an activator, let's get a different color here, an activator comes in. All right, so here's your activator. Oh, I picked pink, but my pink marker is going out. I'll do a darker. There we go. There we go. An activator comes in. So activators, we just says, stabilizes the active site, all right, and makes the enzyme stay functional, all right? So when an activator comes in, notice here, where does it bind? Where do you see it binding? It's binding right here, so it's binding to one of those sites, those allosteric sites that are not the active site. Once an activator binds to one of those sites, it stabilizes all of these active sites so that they can't change shape. So therefore, these active sites stay functional. They don't change shape. And so they stay functional here. So that's what an activator does. So that's what the first part means, is it stabilizes the active site. Let's go through an example, though, of actual molecules that go through this process. So I'm going to write some things over here in the margin. Okay, so over here, let's write, so let's say, oops, this is an enzyme that helps break down glucose. So if we relate this, so let's go back to the ATP cycle right here. So energy from the breakdown of glucose, that's that itself is an exergonic reaction. Remember, releases lots of energy, and that's the energy used to put the phosphates back onto ADP to make ATP. And then we break it down, and then we rebuild it, and so on. So we're talking about breaking down glucose. Energy is released, and we're going to make that energy is used to make ATP. So going back to this, all right, so it's an enzyme that helps break down glucose. So let's put a couple bullets here. If ATP production lags behind its use, what's going to happen? So, meaning that if you're using it more than you make it. All right, so let's go back to that ATP cycle, and let's just look at how that works. So, ATP production lags behind its use. So, therefore, it's going to be used up more than it's being made. All right, so this side is happening more than this side. So what's going to accumulate? You're going to start accumulating ADP, because this is breaking down faster than you can remake it and convert it back to ATP. So we're going to write here that ADP accumulates. Okay. So, what do we want to happen? We want to speed up this side, all right? So we need to make more ATP, all right, because we're using it more and then we're making it, so we want to speed that part up and start making ATP. So let's write here that we need to make more ATP. All right. So what do we need to make more ATP? What needs to happen? Well, what does make more ATP? Where do we get the energy from that? Let's go back here. Where do we get the energy from? From the breakdown of glucose, right? So we need to break down more glucose to get energy so that we can get this process going, all right, and start making more ATP. So let's write that. All right, so what do we need? We need to break down more glucose. To provide energy to make ATP. Right? All right, and let's say that this enzyme in our picture here, with these 4 subunits, this is one big enzyme. Let's say this is an enzyme that helps this process, that helps break down glucose to get energy to make ATP. So if we have, we want this enzyme to work then, all right? Because we want this enzyme to break down glucose to get ATP because we are lagging behind. And so therefore, I'm going to write here that we need this enzyme to work. All right, we want it to work, so we want to make this happen, all right, this breakdown of glucose. So therefore, what happens is we want an activator that is going to stabilize this enzyme so it works, so that it can work to break down glucose. So you know what stabilizes it? If we go back here, what accumulated? ADP, right? So in the cells, the cell can't see, oh, we have a, we need to speed this up because this reaction's happening more. What it does know is that this ADP is accumulating, and this ADP can actually act as an activator. So therefore, right here in my example, the activator is ADP. So when ADP starts to accumulate, it binds to the enzyme and therefore stabilizes it in the active form, and therefore this enzyme is going to work, and we want it to work, and because we have ADP accumulating. So the actual molecule that's accumulating is acting as the activator. So I'm going to put this in the same color here. ADP accumulates, all right? And so down here, let's write this next bullet here. ADP acts as an activator. So this enzyme works. And so, color code all of that, okay? So let's write up here a little something. Okay, so right here, we're going to write what ADP is doing. ADP stabilizes the enzyme. So glucose can be broken down to release energy to make ATP. So it's going to work, it's going to break down the glucose, energy is going to be released, and we're going to start making ATP. So, that's the first part of this. So now let's say that energy or ATP is being made, and let's say that its use slows down. So now we're in a situation where it's being made faster because we're using it fast. So it's being made faster, and glucose is being broken down, providing that energy to make ATP. But now this side starts to slow down. So you start using ATP less, all right? So let's say at first you were running, all right? So you're exercising, breaking down a lot of ATP faster than you could make it, and so therefore the enzymes are activated here to break down more glucose so that you can get more energy to make ATP. But now you're done running, and now you're lying on the couch and you don't need as much energy, and so therefore you're not breaking down ATP as much, and so this part starts slowing down, but you're still got this one going fast, all right? So now we're imbalanced again, and so now what we want to do is slow the making of ATP down. So let's go back to this and write that. So that's going to be the second part of this picture here. So let's write here, if ATP is being made faster than it's being used, what's going to happen? So what's going to accumulate now? So it's being made faster, so this is going on faster than this. What's going to accumulate? ATP is going to accumulate. So therefore, let's write ATP accumulates. So what do we want to happen? We want to slow the process down of making ATP. So we want to slow this part down, all right, because we are not breaking down as much, so we don't need to make as much. And so, so we need to stop making ATP. Or slow the making of ATP. So, how do we do that? So, in terms of this enzyme, remember this enzyme speeds up the breaking down of glucose. And so, therefore, what we want to do is, with that enzyme, is we want to slow the breaking down of glucose so that we're not providing energy to make ATP. And so therefore, if you don't have the energy source, then the process of making ATP will slow down. And so, so let's write here that we need to stop breaking down glucose for energy to make ATP. And the reason why is we already have enough. So what we want to do is make this enzyme so it doesn't work. All right, and so what happens is ADP is going to stop accumulating, and so ADP will come off to be used to make ATP. So the ADP that was once in excess is now going to be used here to make ATP because we're making this ATP too fast, too much. And so therefore, an ATP starts to accumulate, and so what happens is ATP acts as an inhibitor. Acts as an inhibitor. So let's look at this picture here. I'm going to get a different color here. I'm going to pick blue. And so now what's going to happen? So ADP is going to be used up to make ATP. And so then ATP accumulates. And so ATP is going to act as an inhibitor. So the inhibitor is ATP. So now look at what happens here is ATP, when it binds to that allosteric site here, it stabilizes the enzyme in the inactive form. Sorry, and so so ATP. I'm going to put here ATP acts as an inhibitor. I'm going to put here that's the ATP in that color, and let's write a few things about this. So let's say ATP binds and changes the shape of the active site and the enzyme, I'll just abbreviate, cannot break down glucose. To write small to get in here. All right, so it's in the inactive form. And so, and that's because, again, let's get the active site color here. The active site is not functional. All right, so therefore the molecule cannot get to the active site. Okay, and so this is called an allosteric enzyme. So allosteric enzymes oscillate back and forth between the active form and the inactive form, depending upon if there's an activator present or an inhibitor. And so the ATP, when it starts to accumulate, that says, hey, we have enough ATP, so it binds there, stops breaking down glucose, we no longer make any more ATP. Then what happens when you start using up ATP? You're going to start breaking it down into ADP. Eventually, this will come off and you'll use this ATP for energy. And then if ADP accumulates, it'll bind and stabilize it in the active form. So it goes back and forth, oscillating between active and inactive forms. Okay, so let's turn the page and take a look at this next picture that shows one other thing with this allosteric enzyme, and that is what a substrate can do. So a substrate can make the enzyme stabilize it in the active form by binding to it. So here's your substrate right here. So here's your inactive form. If the substrate binds, this is the active site right here. If the substrate binds to the active site, remember there were 4 active sites. Let's keep that same color here. So this was the active site, active site, active site, and active site. So what cooperativity means is that if the one substrate binds to one of the active sites, then it stabilizes that enzyme in the active form. And it doesn't matter if there's already a substrate bound to it. It doesn't matter if an inhibitor binds or not. And so it's going to stay in the active form. And so let's write that here. So let's write this, all right? Binding of one substrate molecule to the active site of just one subunit locks all the subunits in the active form. Conformation shape. And so, so that's called cooperativity. And so that is another thing. So not only does an activator stabilize it in the active form, but a substrate can stabilize in an active form as well. All right, so both of those can happen. All right, and that is allosteric regulation of an enzyme. And the last thing, pulling it all together with reactions and enzymes and how they work, is this thing called feedback inhibition. So what happens here is the end product of a metabolic pathway shuts down the pathway by binding to the allosteric site of an enzyme. So let's talk about what this means. All right, so before we go on to this bullet here, which just reminded me that I didn't go back and do this bullet up here about cooperativity. So we just talked about cooperativity, and so let's do this. So one substrate triggers a shape change in other active sites which increases the catalytic activity. So what does that mean? It means that the substrate stabilizes the active site's shape and therefore increases the enzyme being able to catalyze reactions. And so therefore it is in the active form and can catalyze more reactions or increases the catalytic activity. All right, so sorry about that. So let's go back to the feedback inhibition where we have this metabolic pathway. So let's talk about what that is. All right, so the overall reaction here is threonine, which is an amino acid, is going to be converted to isoleucine. All right, so that's the overall reaction. So oftentimes we say the reaction is, we're converting threonine to isoleucine. Both amino acids. And this arrow here represents a pathway. So there's really several smaller steps that need to happen. And therefore, threonine is converted first to intermediate A, and that's catalyzed by enzyme 1. So that reaction is sped up by that enzyme. Then A is converted to B, and that's catalyzed by yet another enzyme, enzyme 2. You see the picture, B to C, catalyzed by a third enzyme, C to D, catalyzed by a fourth enzyme, and then eventually D to isoleucine, which is catalyzed by a fifth enzyme. So just to convert from this reaction, there are a series of reactions that each are catalyzed by these 5 different enzymes. All right? So that's what is meant by a metabolic pathway. So what happens is threonine binds to the enzyme here. And here's just the name of the enzyme. Notice it ends in ACE. You don't need to memorize that enzyme or anything like that, but it ends in ACE. And so it binds to the active site here. So this is the substrate. Binds to the active site, gets converted to A, B, C, D, and so on, and eventually you get to isoleucine. And so what happens is what if, so let's write here an if. So this, if threonine starts to accumulate, or sorry, not threonine, isoleucine starts to accumulate, what happens is it will inhibit the enzyme at the start of the pathway, and so therefore stop the pathway. So let's say that you need isoleucine, so you keep making isoleucine, and then all of a sudden you don't need it, and it begins to accumulate and build up. So what happens is, if we go over here, the isoleucine, let's do another color here. So this is isoleucine here. The isoleucine, if you notice here, binds to the allosteric site. So this is what the enzyme actually looks like. And so you have the enzyme that has, this is the active site right here. But then it also has, let's do, I don't know, purple. It also has this allosteric site. And so if isoleucine binds to the allosteric site, what happens is the active site changes shape. All right, so the active site is no longer able to catalyze the conversion of threonine to intermediate A, because if isoleucine binds to that, it changes the shape, and therefore threonine is not going to be able to fit into the active site anymore, which means it's not going to be converted to intermediate A. And if it's not converted to intermediate A, can the rest of the reactions happen? Absolutely not. And so therefore, when this accumulates, it binds to that and shuts off the pathway by inhibiting the enzyme at the first reaction. And so therefore, what the purpose of that is so that it's not wasteful, because if you don't need isoleucine, it'd be really wasteful to continue this. And so therefore, as you don't use it, and it inhibits that first enzyme, and you're not making any of the intermediates there, and so therefore it's more energy efficient and more efficient for the cell to conserve resources by shutting it off at the first reaction. And that's why the enzyme 1, this is inactivated, and you don't inactivate enzyme 4, because then you go through all this process here and you don't need to. So the first part of the pathway is shut down. So let's write up here. Let's write up here that it prevents the wasting of chemical resources and increases the efficiency of the cell. Okay? And then what happens is, as you start to need isoleucine, what's going to happen is your cells are going to use up all the isoleucine, and then it will actually use the isoleucine that is attached to the enzyme. So that's what it says here. Isoleucine is used by the cell, so it can detach, and then the enzyme goes back to normal, and then the process can happen and you can make isoleucine. Then as it starts to build up, it inhibits the enzyme, doesn't use it, can't be used, and so this process doesn't happen. As you start to use isoleucine, you use it away and take it off of the allosteric site, goes back to normal, and so on and so forth. And so therefore, we are regulating the production of isoleucine by regulating the enzyme at the first step here. And so this can be called negative feedback as well, all right? So, or feedback inhibition. And that is the end.